Protein sequencing is the identification of the amino acid sequence of a polypeptide chain or a protein. Already we found that the different polypeptide chains in the protein are initially separated. This is by breaking down the disulfide bonds between them. Thereafter, each of the polypeptide chain is hydrolyzed into a series of fragments using different sets of acids, enzymes, etc. And thereafter, each of the smaller peptide we got is sequenced. That is the general mechanism involved. And thereafter, by means of identifying the overlapping sequences, we find out the complete sequence of the polypeptide chain. A process called N-terminal analysis is very much important in identifying the N-terminal residue. What is the N-terminal residue? It is the amino acid at the N-terminal, the amino terminal of the polypeptide chain. The importance of this process is that once we identify the N-terminal amino acid, we can find out how many polypeptide chains are there in that protein. Point 1. Point 2. Sequencing of the amino acids of a smaller peptide is actually done by identifying the N-terminal amino acids. For example, take the case of insulin. Insulin, when studied by the researchers, was found to have equal amounts of glycine and phenylalanine at the amino terminals. What does it mean? There is a polypeptide chain consisting of or containing glycine at the amino terminal side, and there is another polypeptide chain containing phenylalanine on the amino terminal side. It means insulin consists of two polypeptide chains. So that much important is the N-terminal analysis. And how can we carry out N-terminal analysis? There are three methods. There is the Sanger's method, and of course this method is modified later. Then there is the Edman degradation, and also there is the dinyl chloride method. Let us go to these methods one by one. Frederick Sanger. Frederick Sanger was a great scientist. He won Nobel Prize 2 times. First time Nobel Prize in Chemistry in 1958 for his studies on the sequencing of proteins. Actually, Frederick Sanger analyzed the amino acid sequence of insulin, and later he was awarded a second Nobel Prize in Chemistry in 1980 for developing methods for the sequencing of DNA. And I think he passed away in 1913. What was actually Sanger's method? Various samples of the protein are broken into fragments using acids or enzymes. You see, the same protein solution is divided into a few test tubes. Into each of the test tube, a different acid or a protease enzyme is added so that the same protein molecule is hydrolyzed into different fragments in different test tubes. Thereafter, the fragments of each test tube are separated by means of chromatography, etc. And each of the fragment is further broken down with the different sets of acids and enzymes. This will help to identify the overlapping sections of any peptide sequence. And finally, these smaller fragments, overlapping fragments, are completely digested and individual amino acids are separated by means of chromatography. and thereafter each of these amino acids is identified. In chromatography, based on the speed of movement, based on the solute front, based on a value called RF value, we can identify amino acids. And using the overlapping nature, the complete sequence of amino acids is reconstructed on a paper. That is how we are doing it theoretically. And how did Sanger identify the N-terminal amino acid? There's a reagent 2,4-dinitropluorobenzene. Look here. This is carbon number 1, 1-fluoro, 2,4-dinitrobenzene. That's what's written here. 1-fluoro, 2,4-dinitrobenzene. So this peptide, we say an oligopeptide, is allowed to react with 1-fluoro-2,4-dinitrobenzene, and this fluorine atom reacts with this hydrogen of the amino group, releasing HF. And what happens? This carbon is now engaged in bond formation with the amino nitrogen of the oligopeptide. And this DNP amino acid peptide is now hydrolyzed using acid. You know what happens? The DNP amino acid is hydrolyzed from the rest of the peptide chain. What is DNP? Dinitrophenyl. So dinitrophenyl amino acid is actually the amino acid at the amino terminal of the oligopeptide. That amino acid along with the DNP group is hydrolyzed from the rest of the polypeptide chain or the oligopeptide. And then this DNP amino acid is easily identified using paper chromatography. And this is a paper chromatogram actually developed by Sanger in 1950s. This is the paper. Here is the solvent front. Here is the origin. These are the sample wells. Here he applied 5 samples, and these amino acids are moving at different rates. He identified different amino acids. You can see the amino acids he identified using standards. This is an actual chromatogram. This is the notebook of Frederick Sanger in 1953. You can see here, December 1953. He was working on insulin. He identified that insulin consists of 2 polypeptide chains, and he identified many of the amino acids, but not all. Many of the amino acids, see here a gap is there because he could not identify the amino acids there. And here he established one of the disulfide bond very clearly. And here there is a cysteine on one chain. There are 3 cysteines on the other chain. At this moment, he could not identify the position of the disulfide bridge. That is why he has not completed it. Remember, this is the true nature, true quality of a researcher. A researcher is writing down every observations, every work in a notebook. Nowadays also researchers are following this way of practice. And later, Edman modified this Sanger's method. Instead of the 1-fluoro-2,4-dinitrobenzene, Edman used another reagent, phenyl isothiocyanate, PITC. Let us see the reaction first. This is PITC, phenyl isothiocyanate. This PITC is reacted with a polypeptide chain or an oligopeptide chain. Let us say it has n number of amino acids. This is the N-terminal residue. This PITC reacts with this amino nitrogen of the oligopeptide chain. You see, PITC polypeptide is produced. Phenyl isothiocyanate polypeptide is produced. This polypeptide is then hydrolyzed using trifluoroacetic acid. Trifluoroacetic acid, what happens, the amino terminal amino acid along with the PTH is removed from there by means of hydrolysis, and the rest of the polypeptide chain also is there. What is PTH? Phenyl thiohydantoin. This is now called phenyl thiohydantoin amino acid. This amino acid was actually at the amino terminal end of this polypeptide chain, and the remaining part of the polypeptide chain is there. It contains N-1 residues. Now this PTH amino acid can be easily identified using paper chromatography, using different sets of standards. This method is advantageous over Sanger's method. You know why? Sanger's method has a problem. After each round of hydrolytic removal of the N-terminal amino acid, we need to add fresh protein sample. You see, advantage of Edman degradation over Sanger's method is that this Edman degradation does not require addition of new protein sample after the removal of each amino terminal amino acid. Not only that, this method is now automated, which means now machines are invented which are controlled using a computer. You need just to add less than 1 microgram of the peptide solution along with the necessary reagents, and you give the necessary commands using the computer. The machine will do everything by itself. Usually we use oligopeptides not more than 50 amino acids long. You know why? By the time 50 amino acids of a chain are analyzed, the solution becomes more and more impure. So we are initially fragmenting the proteins using different sets of amino acids and different sets of acids and enzymes, so that we may get oligopeptides containing not more than 50 amino acids. And then each of the polypeptide chain is analyzed using this automated Edman degradation technique. Trypsin and chymotrypsin are two important protein digesting enzymes used for this work. You know what's their importance? Because trypsin and chymotrypsin are highly selective. Trypsin hydrolyzes carboxyl side of lysine or arginine residues. Just imagine this is a polypeptide chain. This is the amino terminal. This is the carboxyl terminal. Imagine there is a lysine here. There is a lysine. And this is another amino acid. We call it X. If you are adding trypsin, trypsin will break down here. It will break the polypeptide chain at this point, where on the carboxyl side of lysine. On the other hand, if there is an arginine present here, then trypsin will also break this polypeptide chain on the carboxyl side of arginine. Arginine is ARG. Similarly, chymotrypsin also is so much specific in that it hydrolyzes the carboxyl side of tyrosine, tryptophan and phenylalanine residues. Just imagine this polypeptide chain was attacked by trypsin. It contained an arginine residue and a lysine residue. How many fragments will be formed by this hydrolysis in that test tube? There is a long polypeptide chain. There are 2 points of hydrolysis, and how many fragments will be formed in that test tube? Fragment 1, fragment 2 and fragment 3. There will be 3 fragments formed, because if a long string is cut at 2 points, you know that 3 fragments will be produced. So this is how we break down the polypeptide chains and we identify the N-terminal amino acids. We already found this Edman degradation. Next one is dancyl chloride method for the digestion of or for the tagging of N-terminal amino acid. We will see it in the next slide.